Okay, good morning, everyone. Am I audible?
Yes, Raj.
Okay, fantastic. Um, so last few lectures, we've been talking about some of the concepts involved in thinking about developmental biology. We've talked so far about ways that cells can reorganize themselves. We've thought a little bit about signaling molecules and how gradients are set up in determining pattern. We've talked about gene networks and how gene networks are interlinked to make robust patterns in the nucleus. And last lecture we talked about stem cells and thinking about signaling and networks, transcription factor networks to regulate pluripotency. So today I'm going to try and wrap up this, my chunk of lectures. These are meant to be concepts in developmental biology. I'm going to wrap everything up by talking about ways and methods that we can use to kind of understand development. And so these are essentially developmental methods, so embryological methods, and not so much genetic. And so Tina in the next batch of lectures from next week, will talk about the power of genetics in understanding developmental biology, and she'll be focusing primarily on fruit flies. But today I'll be talking about other methods that you can use to understand development. I should say, if anyone has any questions on what I've lectured so far, please go ahead and ask. Okay, and if not, I'll start. So, ways to understand development. And so what I want to talk about today is kind of almost new ways of thinking about development, some old ways, and kind of where I think developmental biology studies are heading to. So I'll start this by thinking about lineage and potency. And so, of course, all of you remember that potency reflects what a particular cell is able to give rise to. And this is intimately linked up to the lineage of a cell, so what the descendants of that cell would be. And I want to talk a little bit about methodologies that we can use to understand what kind of cells, what kind of daughters a particular precursor cell will give rise to. Before I go on, does anyone want to hazard a guess on how we can understand lineage? Am I audible? Can you hear me? Yeah.
Yes.
Yes, Raj, can it be with the help of fate maps?
Fantastic. Yeah, exactly. And so let's, do you have an idea of how to generate a fate map?
Raj, how they used so in the previous days, they used to use some vital stain and stain a particular part of the embryo and then find out which part it becomes in the future.
Yeah.
And from that, they advanced to you incorporating radionucleotides into one of the cells or a particular region of the embryo. And later they track which part of the adult organism has radioactivity.
Okay, fantastic. You could probably give my lecture then, Shubha. Thank you. Yeah, that's exactly right. I mean, the easiest way is to kind of watch cells. Okay, and so if your model system is tractable enough, you can just look at how the cells are developing. And so C. elegans is a classic case. So the C. elegans embryo is transparent, and the cells always give rise to fixed descendants. And so a classic paper from 1983 by John Solsten basically traced each descendant of all of these cells by light microscopy. Light microscopy, some labeling, but not much. But just asking where they can track these cells. The C. elegans embryo at these kind of stages only has 959 somatic cells. And so it's relatively easy to understand the lineage of a particular cell. And then so this, just by looking, Solsten was able to just trace every single cell in the C. elegans embryo with the C. elegans is kind of very handy for this. It's a fixed lineage, so the development is regulative, and so that was fantastic. And this this kind of highlighted the power of C. elegans. The next way to track cells is, as Shubha mentioned, by vital dye staining. And these are experiments done in amphibians from almost 100 years ago. And that what Vogt did was to soak these chips of agar with particular vital dyes and put them on the neural, just on the ectoderm of the embryo. And in this way, he was able to show that various bits of the ectoderm gave rise to various bits of the brain, of the central nervous system. So this dye here, a dye placed in this one position, would give rise to the forebrain, whereas a dye placed further caudally would give rise to more hindbrain type characters. And this kind of analysis was very powerful and popular. There's also putting in radiolabel, particularly tritiated thymidine. Tritiated thymidine would incorporate into the DNA, and then all the nuclei descended from that injected blastomere would be labeled with tritiated thymidine. You can reveal where the tritiated thymidine is just by simply sectioning and putting an emulsion of silver on top of the embryo. Where there's some radioactivity, the silver will deposit, leaving a grain wherever you see tritiated thymidine. So these are some of the ways that were used in the past. Along the same lines as tritiated thymidine, David Weisblatt, who's still working, he's in the University of California in Berkeley, has done a fantastic fate map of the leech embryo. And what he's been doing is using lots of different techniques. He started off using tritiated thymidine in the late 80s, and now is using various dyes. And so in this particular paper I'm showing here, he's looking. He's trying to trace these M cells, these mesoblasts, and asking what they give rise to. And here, all he's doing is injecting the mesoblasts with a plasmid that encodes a GFP reporter, and then asking where this ends up, where these cells end up. And in this way, he's seeing that these particular mesoblasts end up in a segmented fashion. Probably in the nerve cord of the leech embryo. So these kind of lineage labeling experiments are very straightforward, very powerful. They're giving you an idea of what the potency of each cell is. Yep, was that a question? Atal, did you have a question, or was that by mistake?
That was by mistake, Raj.
Okay, all right. So the other another way that we can kind of understand lineage is by labeling the cells genetically. And so I've shown you this kind of scheme already when we talked about gene regulation and enhancers. So you can have an enhancer drive a particular gene in a subset of cells, and this will give you some idea of what these cells will give rise to. For example, here, what you're seeing in E is this gene, MEF2C. MEF2C is driving a reporter gene, LAC-Z, beta-galactosidase. It will cause a blue precipitation in particular conditions, and what this shows is that this group of cells here will give rise to the most anterior neural cells in the later embryo. Of course, so this of course is driven by a particular enhancer. Here they've knocked in, so they've taken an intronic enhancer to drive LAC-Z. And of course, enhancers are active at particular stages in development. They're not always active. And so this is not quite a lineage tracing. It's just showing you these cells are active at this point. Later on, this enhancer is active in neural crest. And so you can see neural crest here. Most of you know what neural crest is. Do you? Do I need to explain neural crest?
Yes, Raj, please explain.
Okay, all right. My plan was to explain neural crest in about a month's time. Neural crest is a migratory population of cells that emanate from the top of the neural tube, and they'll give rise to, and I'll show you this in a couple of slides. And they'll give rise to things like the jaw, the bones of the face, various sensory neurons, pigmented cells, and things like that. Okay, I'll show you this in a few slides. Okay, but here you can see the neural crest has come from particular streams in the brain. It's filling up what are called the branchial arches, and so these are the precursors for the jaw musculature, the jaws and the face. And as the face is forming, you can see neural crest labeled here. And these end up kind of in the jaws in various axonal tracts which are lined with neural crest cells. So these neural crests will also form some of the glia as well, and so on. So this is labeling cells genetically using an enhancer that's driving water. So as I said, this is so enhancers are dynamic, and so these cells we know are not neural crest cells, but this enhancer drives expression in neural crest a little bit later. One way that we could label them permanently is using this Cree-Lock system. So let me ask you if anyone knows the Cree-Lock system. Anyone? No? Okay. So, so these are Cree and locks. So it's called the Cree-Lock system. These are individual transgenic mice, okay? And so the Cree protein is known to cleave these LOX-P sites in bacteria. And so what's being done is that a driver that so mice have been made that express Cree, other mice that express lock sites, and here now we can get conditional expression of particular genes. So when a Cree mouse and a Lox mouse are mated together, a recombination happens around the Lox site, so you now have expression. So the condition is if Cree is present, if Lox is present, gene is on or gene is off, okay, depending on how you're setting up the construct. In this case, what you can do is put a transcriptional stops sequence in between the Lox sites. And so what one mouse will have is an enhancer driving Cree. Yeah, so don't worry about this GFP and this IRES. So this enhancer is driving Cree in a particular subset of cells. Let's not worry about this ER either at the moment. The reporter contains a strong promoter, which is ubiquitous, but downstream of this strong promoter is a transcriptional stop that's flanked by 2 LOXP sites. And these will, and beyond this is a reporter construct, and a reporter gene here it's LAC-Z, it could be GFP, it could be any readout for the activity of this promoter. In general, the promoter is, so because of this stop cassette, beta-galactosidase is silent, but when this mouse is mated with this Cree-expressing mouse, in these Cree-expressing cells, you get recombination and this stop cassette is recombined out. And so now in those cells, LAC-Z is expressed. And LAC-Z is, of course, beta-galactosidase, which you can color blue using particular chemicals. And so in this way, you can start finding the progeny of all of these cells, the Cree-expressing cells, and what they've given rise to. And here, what this experiment is looking at this particular Cree line, LGR5 Cree, which is a marker for intestinal stem cells, and they're finding that all the progeny, and so generally LGR5 is expressed in just the putative intestinal stem cells, which are down here somewhere. But in this, but the progeny, you can see now span the whole of these intestinal villi. Okay, so that's one way that lineage can be traced genetically. So if you have any questions here, please ask them. Okay.
Raj, I have a question.
Yeah.
I was just wondering, so why do we need a conditional gene expression system here? Because we would want to see that lineage throughout the development, right?
Yeah. So so we need the conditional system here because we know that. So you can see my cursor, yeah.
Yeah.
Yeah. So we know that LGR5 is only expressed in intestinal stem cells, and intestinal stem cells are. Oops. Sorry. Intestinal stem cells are only found at the base of the villus here.
Okay.
And so, and we know that. So if we were labeling with just an LGR5 beta-galactosidase, only these cells would be showing blue staining. Okay, but we're interested in the progeny of these cells. Where do these cells end up? And so what we're doing is causing a recombination in these cells, and then asking where those cells that had once expressed LGR5 are going. Yeah. So there's kind of this nuance of now and progeny. Yeah. Is that kind of clearish?
So when we add in the Cree system, even the progeny, I mean, both the parental cell and the progeny will both express the marker.
Yeah, the progeny will eventually express the marker. Yeah. Okay. even though LGR5 is no longer expressed there, the LGR5 Cre was once expressed in those cells, okay? And that's sufficient for this recombination to happen, activating LAC-Z, yeah?
Okay, yeah, thank you.
Okay. So this kind of Cre-mediated recombination has been further finessed. You can, of course, add in multiple combinations of fluorescent proteins, for example, and have different types of recombination to make different colors. And so, so for example, here is what's called a rainbow construct. You have GFP and YFP, RFP and CFP, so blue fluorescence, red fluorescent, yellow and green fluorescence, all put into this construct with different configurations of LOX-P sites. And what these LOX-P sites, if two LOX-P sites are facing each other, then you get an inversion. If two LOX-P sites are facing in the same direction, you get a recombination. And so you can see that there's a possibility of many types of inversions and recombinations in this construct upon Cre activation that will give you different colors. And so here, for example, if you recombine out this unit, the blue and red unit, you're left with green. YFP is facing the other way, and so that won't be expressed. If you recombine out this unit, but flip this unit, you get YFP and so on. And this is kind of stochastic. The recombination will happen once, and then all the progeny of that cell where this recombination has happened will have different colors. Okay, and in this way you can start, if you have a tandem array of this construct, you can start getting different copies of different fluorescent proteins in different cells. And so in this way you can start getting a rainbow of fluorescent proteins, all in Cre-expressing cells. And so now within a Cre-expressing domain, you can start labeling individual cells and then asking for where the individual progeny end up. And so in this way, you can see this figure down here. This is neurons, and so you're labeling the cell bodies with a particular Cre. This is skin here, and so again, these are different strata. These are epidermal cells, all labeled with one particular Cre, but now you're looking at individual cells and asking where they've ended up. This is called confetti or rainbow. Here, Alex Shear's group had made a zebra bow, zebrafish. And again, you can see what you're able to do is start getting individual cells labeled in different colors. And if you can then track these cells in real time, you can understand then where each progeny is ending up and and what the lineages of individual cells at particular times. And so this kind of analysis, this high resolution analysis, is incredibly powerful. Okay. Any questions here? Good.
Raj?
Hi.
I would like to know how long this lineage tracing can be done. Is there an extent of this much cell division? Say like up to 10 cell divisions or something, or it is like lifelong?
It's lifelong. So these are recombinations that are happening in the genome. And so as long as the, sorry, let's go back. so you notice there's a promoter here that's driving all of these the expression of these cassettes downstream. As long as this promoter is active, then it's lifelong, okay? Or it's as long as the promoter is active for. So it depends on this supposedly constitutive reporter that's driving the expression of the reporter genes, constitutive enhancer that's driving the expression of the reporter genes, okay? And so as long as that's active, then these can be seen. And it doesn't depend on cell division. It's not being diluted as the cell divides, okay? Because this is tagging, this is a recombination that's happening within the genome. Is that kind of clear?
Yeah, yes.
Yeah.
Thank you.
Okay. So this is then another way that cells can be labeled genetically to understand lineage. Another way is just by putting in new cells. And so here what I'm showing is some classic experiments by Nicole Ledoran. These are quail cells that are being transplanted into chick embryos. This, I think, would some of you have covered this during your undergrad or master's courses? Anyone? No? Okay. So quail chick transplants have given us a ton of information. And so everyone knows what a quail is. You know what a chicken is. They're both from the same general family. They're galliforms, and they kind of develop simultaneously. They develop with the same timing, and there's not too much difference in the size of the embryos. Quail cells and chick cells are different though. So quail cells are marked by a nucleolus, whereas chicken cells don't have a nucleolus. And so what you can do is then replace part of the chicken embryo for the corresponding quail part, and then ask what that part gives rise to just by looking for the presence of nucleoli. This technique was first used by Nicole Ledoran a long time ago. She finessed it by identifying an antibody that was specific to quail cells and not to chick cells. And in this way, she could start understanding where groups of cells were going. So for example, here ectoderm from this region is being transplanted. Here, kind of neural crest, so the dorsal part of the neural tube is being transplanted, and these are giving rise to particular structures. And so she found out that when you transplant this ectoderm outside here, you get these kind of this kind of lineage. When you transplant neural crest, you get this kind of lineage here. And so this is neural crest cells here. And doing this systematically by taking the corresponding bits of the neural tube, for example, along the whole rostral caudal axis of the embryo, she was able to then map out where each individual neural crest stream was going to. And so she found out just purely by quail to chick transplants that anterior neural crest would give rise to kind of the bones on the around the upper skull, for example. This neural crest here, this red neural crest, will start giving rise to the jaw bones and the upper jaw bones, the maxilla and mandible, and so on. Crest that was coming from here would give rise to... Hi?
Sorry to interrupt you, but how did she track where the cells are going after the transplant?
Yeah, so all she did was so all she did was kind of fix them at particular time points. So she didn't track them. She did the transplant maybe, so this stage is about 2 days after incubation, and then put the experimented embryo back into the incubator and left it for 9, 10 to 14 days. And then after 14 days, she took the embryo out. After 14 days, the embryo looks kind of like this, okay? It's 12 days here. And she just sectioned it and then looked either for nucleoli when she didn't have the antibody, or she stained this for the quail cells when she generated the antibody. And so it's purely that, okay? And so of course these studies were done over the course of about 20, 25 years to dissect out where each individual, where different bits of the neural tube were ending up, and which bones were being generated from which pieces of neural crest. And so that's basically how she did it. Okay.
Okay.
Yeah. So definitely this is not a PhD project. Okay, this is something that took her a long, long time to do. The quail, it's a side, an aside here. The quail chick transplant strategy is really, really powerful. And so some experiments that she did do is to ask, well, if you put a quail neural tube into a chick and let it be born, is the resulting animal quail-like or chick-like? And so quails and chicks have different behaviors. Quails are more aggressive. Chicks are more passive. Quails are less prone to imprinting. Chicks are. Chicks will peck more. Quails don't peck so much. And so using this way, she found that if she put a quail neural tube into a chicken embryo and allow it to be born, the resulting chicken, and it was a chicken, it just had a quail brain, had characteristics which are more quail-like. This chicken was more aggressive. It was less prone to imprinting, and it had and it didn't peck so much either. So that was kind of interesting. Extending these studies, she found that neural crest as well also gives rise to pigmented cells, and so she could recreate the pigmentation of a quail embryo of a quail in a chicken as well. So just very, very interesting experiments. Okay. So this is so these transplants are done by hand. Okay. What you're doing is just cutting out the neural tube and replacing it for the corresponding piece in the host. Okay. You can do something almost similar, but not quite, which is called chimera analysis in mammalian embryos. And so here you can take different parts of an embryo or different kind of ES cells, for example, and place them back into a blastocyst and then ask what these cells are able to give rise to. And so, for example, here, putting an embryonic cell into an embryo host. So here the donor cells are GFP labeled, the host is not. You can see that if you do this embryo to embryo kind of injection, you get fairly good distribution of cells everywhere. And that tells you that these cells are able to give rise to any part of the embryo. Okay, ES cells to embryo implantation, as you'd expect, because the ES cells were derived from only the embryo and not the extraembryonic tissue or the trophoblast. ES cells would only give rise to the embryo proper. And then trophoblast stem cells will, when put into an embryo, again, will only give rise to trophoblast cells. Okay, and so this kind of analysis is very nice as well. You can extend this analysis by taking ES cells, for example, for taking ES cells where a particular gene has been knocked out, and then asking, in the absence of this gene, what these cells are able to give rise to. Okay. Any questions here?
Raj, I was just wondering, were these cells taken from the same organism and then they were labeled and then put back in, or was it from another organism?
No, they're all from the same organism. Yeah.
Okay. They were taken out and then they were labeled and then put back in.
Yeah, exactly. Also, yeah, or they may be from a different mouse strain, for example, which is expressing GFP or expressing some label. Is usually what happens. Okay.
Okay.
Yeah.
Thank
you. Yeah. But I mean, maybe what you're getting at is whether this could be done cross species. Yeah, whether this is a mouse embryo, the host is a mouse embryo, whether you could put human ES cells or rat ES cells in.
Yeah, I was wondering if that was.
Yeah.
Maybe it'll develop properly or normally.
Yeah. I don't know. Actually, I do know, but why don't you find out the answer? Okay.
Okay. Yeah. I will. Yeah.
And you tell me. Okay.
Yeah.
Okay. So ex vivo models, and so what we're coming, and so what the mouse experiment kind of tells us is that mice are incredibly powerful, but most lots of bits of development, particularly of the mouse, are hidden. Yeah, so the mouse develops in the mother, and so understanding kind of the morphogenetic processes or the cell rearrangements that happen during the formation of mammalian organs is really difficult. And so recently people are taking kind of ex vivo approaches to understand the development, particularly of mice. But and so many of you have probably heard of organoids. And so we'll talk about organoids in the final 15-20 minutes or so. Okay, so the idea that you could take bits of the embryo or groups of cells and culture them outside is not new, and so I brought this up because this was actually my PhD thesis.
I think. Okay. And that's going to be pretty nice resolution.
Okay, so
it might even have sound, so it's probably recording us right now.
Yeah, it is recording you.
Because I have another little movie of.
Sorry, I didn't realize this would have sound.
Dissecting out some oocytes. Am I on the screen? Yes.
Okay, I didn't know that would have sound. Okay, well, it seems like all I've got is them speaking. So, so what this would have been is dissecting the future epidermis of the Xenopus embryo, and so what you can do is this is the Xenopus embryo. So Xenopus, if as you remember, is an amphibian. The eggs are like this. This is the blastula stage, and in cross section it looks like this. There's a top and a bottom. When you culture the top, it will form ectoderm, and so it will form basically epidermis. The epidermis is ciliated. These little pieces of ectoderm move around. It'll also, the bottom will form endoderm. Endoderm will be yolky. It will form gut tissue eventually, and so that's those are able to be cultured by themselves, and just in a simple salt solution. You can bring ectoderm and endoderm together, put them close together, and at the interface you'll form mesoderm. And this is one way that the idea of mesoderm induction being a signal from endoderm acting on ectoderm was hypothesized. And then if you can replace the endoderm with particular signaling factors. Here it's FGF, also a signaling factor called activin will do this, and you will induce mesoderm in the ectoderm. And so this is kind of a, I think of it as an early organoid culture. You can go further. For example, this is what, after about 2 days of culture, the animal cap, it's called, the future ectodermal piece, will give rise to just a round ball. But when you add in a growth factor here, activin, you'll start to see elongation. When you section these will now have notochord. They'll sometimes have somites on either side of the notochord, showing a very beautiful organization from this structure. Okay, and so this idea that there's this autonomous organization that happens without the need for a larger context is really attractive, and this is shown more clearly when people have taken stem cells and tried to make organoids from those. So as we talked about teratomas last time, and we started talking about the differentiation to various structures of ES cells. So pluripotent stem cells can be differentiated. So once you take them out of the media that maintains pluripotency, you'll trigger differentiation. And so this differentiation does represent development. And so, for example, what you're looking at here is spontaneous differentiation and self-assembly without any steering of the pluripotent stem cells. What you'll get is a teratoma, and this is a teratoma. I was talking about this in the last lecture. These are either testicular or ovarian cancers that have many different cell types. This one is a tumor that contains teeth, which is incredibly disturbing. But by steering differentiation a little bit, so maybe by adding factors that take these stem cells and upon differentiating, move them to a neuronal fate, you can start getting a neuronal organoid. Yeah, and so here you can see two eyes forming. So this is this green protein is RX driving GFP. RX is a transcription factor that at these stages is expressed in the eye. And you can see that here then a very beautiful CNS organoid. You can steer differentiation a little bit more. So what's happening here is a certain cocktail of growth factors that are steering the pluripotent stem cells to adopt an endodermal and intestinal fate, and you start getting an intestinal organoid. And then steering differentiation even more, or even directing differentiation. Here, what's happening is you're adding cocktails of growth factors that are allowing these stem cells, or are telling these stem cells to adopt a hepatocyte fate, and purely a hepatocyte fate. And this is what is being differentiated. And so what the point of this slide is that by allowing stem cells to differentiate, the outcome, you can't really predict the outcome. But once you start adding some of the signaling factors that have been characterized by regular developmental studies, you can start steering differentiation to structures that are important for you. So here, if you're interested in the CNS, for example, you can start generating a CNS organoid, but simply by inhibiting BMP signaling and promoting FGF signaling, which is known to promote neuronal differentiation. Okay. Any questions here before we move on? So the power of this comes from when you start.
I have a question.
Yeah, go on.
So if we have differentiated one, pluripotent stem cell to some organ, like neuronal one,
yeah,
then can we just re-differentiate it to some other organ?
So not generally, yeah. So this is kind of a one-way pathway, yeah. And so once you and generally, and that's true of development as well, yeah. That once you get differentiation, it's quite difficult to go back. I mean, we discussed last
Yeah,
week that, yeah, and so it's possible that you could then take kind of this eye, transfect in those particular cocktail of factors to reinitiate pluripotency, and then differentiate that into an intestinal organoid. So I think that's I think that's very, very possible. Okay.
Yeah.
The caveat here is that when you do that experiment, after being differentiated into the CNS, it's kind of very inefficient to then. It's not inefficient to take it back to pluripotency, but it's more inefficient to then take it to a different fate. This CNS organoid will. So if you differentiate this CNS organoid that's been induced to be pluripotent and differentiate it again, it tends to form a CNS organoid again. Okay, and so there seems to be some memory that's still there. Okay. So, like I said, organoids represent development. There are caveats, of course. So, in many cases, an organ needs much more than a single tissue type. So, for example, here we're thinking about endoderm. If endoderm will give rise to the precursor of the gut forms, but the formation of the gut needs surrounding tissue to instruct it. It needs contributions from things like vascular cells, nerves, particular types of macrophages. It needs a microbiome to be active. Okay, and so when we start thinking about taking these organoids and maybe transplanting them into humans, that may be some way off, because I don't think culturing ex vivo is going to recreate what happens inside the embryo. And that 3D environment is really difficult to recapitulate. Having said that, I mean, many of you kind of are familiar with brain organoids, I'm sure. So here, this is work from Madeleine Lancaster, and what's happening here is taking human ES cells and then differentiating them into neuroectoderm, and then using a particular culture system to start developing cerebral tissue. And what she was able to find is that using this kind of paradigm, she found not very nice. She was able to generate what she calls mini brains. Yeah, and so these are layered structures of cortex, of human cortex, that form layers which resemble the brain very nicely. Okay, and so these are now actively being investigated, because this is a way of getting a handle on human development and human neural development without needing to break any ethical considerations. So brain organoids, so these are cerebral organoids. Hippocampal organoids have been made by Yoshiki Sasai as well. And here you can see very nice hippocampus made from human ES cells, showing very nice CA3 structures here. Okay, and so this is kind of, I think, where developmental biology is probably heading. You know, we're trying to understand. Ultimately, we're trying to understand human development, and this is one way of getting a handle on how humans are different from mice, and transferring a lot of the information we know from mice, chicks, amphibians, zebrafish into humans. Okay, by looking at these kind of organoids. One thing that is mysterious when we're thinking about human development is the very earliest stages, and so what you're looking at here is a comparison between the mouse and the human during the process of implantation, essentially. So a mouse, of course, is very easy to obtain. We know that after compaction and the formation of the blastula, you get some level of differentiation, and then just before implantation, so at these kind of stages, you get more overt differentiation happening. Similar stages in the human are shown here. Okay, you get the human embryos look very similar up until the corresponding stages, but during implantation, something different happens. It's thought that, for example, the symmetry breaking event that sets up the anterior-posterior axis in human happens during implantation, as it does in the mouse. But this kind of stage is incredibly difficult to capture in humans. I mean, at the moment, this has been supposed by looking at primate studies. Okay. What is now becoming more apparent is that using organoid systems, you can start modeling these stages of development, because the organoids will have, you can steer human ES cell differentiation to an extent, culture it in a particular way, and it will start to self-organize into a patterned embryo. And so this is taken from this review from Magdalena Zernicka-Goetz. What you're seeing here is the mouse, and these are human. Okay, the corresponding stage in human. These are mouse organoids, blastoid, epithelial body-derived gastroloid, gastrulating embryo-like structure, for example. And this is the corresponding pattern in human. And you can see here a patterned human ES cell colony, which has very beautiful epiblast-like cells, extraembryonic mesoderm, endoderm-like cells, and then trophoblast cells, all organized in a very beautiful fashion, as the human embryo would be. And so then, now this gives us a handle on kind of understanding human development, particularly at these implantation stages. And we'll talk about this much more when I come back in kind of towards the end of November. Okay, these kind of human gastrulating or gastroloid organoids can be treated with different factors to understand what happens to the pattern of this human gastroloid, and what transcription factors it expresses, whether the signaling molecules that have been determined for other systems are in play here as well, and what the mechanisms of symmetry breaking in the human are. And so this again represents, I think, where kind of the cutting edge of developmental biology is going to, looking at these kind of ex vivo systems to start interrogating human development in a way that's never been done up until now. So I'm going to go back to the idea of lineage tracing to bring up kind of a new for me kind of way of looking at things, and this is single cell trajectories. And so of course when a cell develops, it will express different factors, different transcription factors, different sets of genes in the way during this developmental during ontogeny, during the developmental program. And so understanding what kind of genes are expressed is and understanding whether you can trace a cell molecularly has always been kind of the holy grail of developmental biology. And so we can start doing that by using what's called barcoding. Okay, what barcoding is being able to track clones of cells. Okay, so every cell that comes from a particular mother has a particular genomic profile that you're able to identify. So this is certainly true in humans. So somatic mutations, for example, in cancer, for example, arise, and all of the progeny of those cells that contain those somatic mutations will have the same somatic mutation. And so just by tracking this particular mutation, you can start understanding where those progeny go. So this may be a spontaneous process in human, for example, but you can start inducing this in different animals. Here you can infect various groups of cells with a virus, with a viral library that contains particular barcodes. These barcodes are then integrated, and so a barcode is going to be then a particular sequence of DNA. And so these barcodes are shown here. Okay, these barcodes will integrate into the genome, and then as the cell divides, the barcode, of course, will be carried on into the progeny. We'll go back to what the advantage of this is later on. Like, I mean, you can generate mice that have what's called this Poly-LOX cassette. In the same way that I showed you the rainbow mice, you can start having different combinations of genomic sequences to give you different types of barcodes within the same, within a mouse. And then what you're able to do is at some point take a group of cells, for example, maybe some liver cells or a particular population of neurons, and then do single cell sequencing. So you can sequence each cell, sequence the genome of each cell, and ask whether you can then detect any of these barcodes. And so if you're integrating, if you're, for example, infecting the virus into some part of the dorsal, the caudal neural tube in the dorsal region, and then looking at a particular population of looking at a particular nerve and finding that barcode in that nerve, you can say that this virus infection in this region of the mouse embryo gave rise to these cells. So this is no different from any of the lineage labeling that we've talked about earlier. It gives a slightly higher resolution, I think. But then when you overlay this, if you're going to be doing single cell genomic sequencing, you might as well do single cell RNA transcriptomic sequencing as well. And so now you have kind of a molecular readout of every single cell. You can say that this cell that has this barcode is expressing this battery of genes. And so now we're able to track very beautifully molecular developmental trajectories as well. And in this way, you can start recreating in very high resolution the path a particular cell takes as it differentiates into its various progeny. You know that at a particular stage, it will express this battery of genes, and at a later stage, it will express another battery of genes. But the progeny is all related. And so, of course, this is going to be something that I think will develop more and more in the next few years to become a very powerful tool to interrogate development. Combined with things like organoid cultures, making particular mutations in organoids, we can now start getting a very clear and very high resolution image, high resolution picture of what particular tissues are doing, and we'll start getting a very nice idea of what human embryos will be doing. Because after all, what we're trying to do by looking at mice, by looking at chick, by looking at Drosophila, by looking at fish, is to start understanding human development, I think. And I'll leave you with this very nice video that I found. So when I think about those kind of like human development and what all of the processes are that go into getting any embryo to develop, I actually get goosebumps. I'm not going to show you my arms. They've got, they're covered in goosebumps at the moment. But it just amazing. So that's it. That's all I have today. If there's any questions, please ask.
Hi Raj, this is just a general question.
Yeah.
I was wondering, isn't it ethically difficult to obtain embryonic stem cells from humans?
Yeah, it is. You do need to go through kind of a huge amount of ethical oversight to get these stem cells. But there's kind of an extreme oversight in the use of human embryonic stem cells.
So are they obtained from the embryo, or is it from the umbilical cord stem cells?
No, it's obtained from embryos. So these are kind of embryos, either kind of very early stage abortions generally.
Okay. So can we do the same amount of organoid development using iPSCs also instead of embryonic stem cells?
Yeah. It's possible. And in fact, a lot of the organoid experiments are done with iPSCs.
Okay.
Yeah.
Thank you.
Yeah. But yeah, you can then see the power of then iPSCs if you introduce a particular mutation that's related to some particular neuronal disorder. You can ask what the brain is like in these organoids.
Yeah, like disease modeling.
Yeah, so modeling diseases. Yeah. Okay. Well, if there's no questions, I'll leave it there. On Friday, Sandeep Krishna will talk about mathematically modeling gradients. This is another way of looking at development, which is incredibly powerful. I'll be at the lecture anyway, and so I'll see you then. And then from next week, Tina will talk about the genetic aspect of developmental biology, thinking about Drosophila. Okay, so that's it. Have a lovely day, everyone. Bye.
Thank you. Bye. Thank you.
Bye.
Thank you.
Bye. Thank you.
Bye. Bye.
Thank you. Bye.